Bueno, buenos días y yo creo que vamos a empezar ya. Ya tenemos 60 participantes en el seminario y os doy la bienvenida a todos y a Rosa en castellano y pues muchísimas gracias por aceptar esta invitación y ser la primera conferenciante de nuestro online seminar series of the CBGP. Y voy a cambiar a inglés porque he visto que tenemos bastantes, aparte de que nuestros seminarios son en inglés, tenemos bastantes invitados internacionales, incluyendo varios de los estudiantes de Rosa en Shanghai. Y bueno, pues voy a cambiar al inglés. So, good morning everybody. And thank you for joining us in this first online seminar series of the CBGP. So I'm Antonio Molina, the director of the Center for Plant Biotechnology and Genomics, CBGP. That is a joint center between the Technical University of Madrid, Universidad Politécnica de Madrid, and the National Institute of Agricultural and Food Technology Research, that is the NIA. For me, it's a pleasure to have the opportunity to introduce and to launch this CBGP seminar series that unfortunately will be just online, but fortunately we can reinitiate the seminar program of the CBGP. That is one of our most important activity in science and innovation, and also is a key activity for PhD student and master student. So today we have as a speaker to Rosa Lozano Durán. She is Spanish, but she has been working in Shanghai in the Shanghai Plant Stress Center for the last 5-6 years, and she will be our first speaker today. So yes, I will introduce Rosa. And to do this, I have prepared a very short, a few slides. So as I said, this is the first online seminar program of our center, and just to let you know that that there will be several seminars during the next Fridays, starting today, and we will have already the program set up until February 12th. And I would like to acknowledge the organizer of the seminar, the CBGP seminar committee, and one of the persons that is responsible of this committee is Lucía Jordá, that is also the coordinator of this particular seminar. And I would like also to acknowledge the sponsorization of this seminar by Plant Responding Corporation, that has been supporting our seminar series for the last 5-6 years. So we have today a very great opening because Rosa will give the first talk of this online seminar. So Rosa is, as I mentioned before, is a principal investigator in the Shanghai Center for Plant Stress Biology. The nickname is PSC. That is a center for the Chinese Academy of Science, located in Shanghai, in a very nice place, in the middle of the close to the botanical garden of Shanghai. Is a beautiful place. And I also have remarked here that Rosa, together with other researchers from the PSC and another research center from the Chinese Academy of Science that is located in Beijing, the Institute of Genetic and Developmental Biology, are participating, PSC and EGDB, with the CBGP in this new Center of Excellence that we have founded recently, the Center of Excellence for Plant Environment Interaction, CEPEI. So let me just to introduce briefly what is CEPEI. So CEPEI is a joint center, so you can see all the information of the center in our webpage. And you can see here the picture of when we signed the Memorandum of Understanding to run this center in Beijing and Shanghai. So what we are doing in the frame of the center is just to collaborate in joint projects. And we already just launched the first grant call, and we got the approval of the project that will be financially supported. And these are the projects that will be financially supported in a joint effort between the CBGP, IGDB, and PSC. As you can see, in the majority of the projects, the three centers are participating, and researchers from this center are collaborating. And I would like to acknowledge Rosa and Alberto, and particularly Yang Min-Su and Ki Li, the big effort that they have done just to run this center, and that I guess is a very interesting initiative that will result in science of excellence and also in some innovation and tech transfer activities. So coming back to this CEPEI initiative, we initially was planning to have the first Chinese-Spanish workshop in Madrid on March the 2nd. As you can imagine, this was cancelled due to the coronavirus. It's a shame. And Rosa was one of the speakers. And a few months later, we have the great opportunity to have Rosa again. Oh, now, no, because now this is not cancelled in our seminar series. And I would like to introduce a little bit to Rosa. So as you can see in the education, she did the PhD in the University of Malaga, and after that, or during the PhD, she did several stages as a visiting student in Yale University, and after completing the PhD, she did a postdoctoral stage in the Salisbury lab in the group of Cyril, and from this position in the Salisbury lab, he moved as a junior group leader to the Shanghai Center for Plant Stress Biology, and as I mentioned, she is now a principal investigator there, and also, as you can see in this profile, adjunct professor of the Institute for Plant Protection of the Chinese Academy of Agricultural Science. So I will recommend you to follow the Gemini team in Twitter, because they are very active. You can see here Laura and behind all the Gemini team from Rosa Lab. And as I said, they are very active in a lot of issues related with science and another activity, women in science, and I will suggest you to follow all their activities. So I will not describe too much some of the major achievement of Rosa, but I will suggest you to read some of her more recent papers. One of them published in Cell, another paper published recently in eLife and another journal, and I guess that she will tell us today about some of these achievements. And with this, I will stop the introduction. Rosa, it's really a pleasure to have you here today as the first speaker of this online seminar series of the CBGP, and also as let's say, the first researcher from the CEPEI initiative. And it's an honor for us. Welcome, and please just feel free to start your presentation. Thank you very much.
Okay, can you see my screen okay?
Yes.
Yes,
Perfect.
fantastic.
Perfect.
Thank you. Thank you very much. Thanks a lot, Antonio, for the very nice introduction. And also thanks to the organizing committee, and in particular, Lucia, for the invitation. I'm really excited to have the chance to participate in this seminar series from the CBGP. So I like to begin my presentations with this slide, because I think this slide very nicely summarizes the main research interests of my lab. So what you can see here are two tomato plants, and they are genetically identical, and they have been grown in exactly the same environmental conditions. So that is basically side by side in the growth room. But you can very easily notice that they look very different. So the plant on the right is smaller and it's yellowish. And if you look carefully, you'll notice that the apical leaves are curled. And perhaps more importantly, this plant on the right will never set fruit. So most likely this plant will never give us tomatoes. And the underlying reason for this difference in phenotype, probably you can guess from the title of my talk. So the plant on the right is infected by a virus. And this virus is therefore rewiring plant development and plant physiology in order to promote its own multiplication and spread. So I personally find this fascinating that a virus can do this to its host, but I think you will probably agree with me in that this is even more striking if we take into account genome sizes. Because while the plant that is being infected here has roughly 32,000 genes, the virus has a grand total of 6. And with 6 genes, it is capable of making a plant like this one on the left turn into something like this one on the right. So in a nutshell, in my lab, we are trying to understand how this can happen. So how a virus can infect a plant and manipulate its development and physiology, and how the plant in turn can try to defend itself upon perception of this viral invasion. And for this, we are using mostly one family of viruses, which are Gemini viruses, and more specifically, one particular species that we use as a model, and that is tomato leaf virus, or TYLCV. So you can see here a representation of the genome of TYLCV. So you see it's a circular single-stranded DNA genome, and it contains bidirectional and partially overlapping open reading frames, a total of 6, as I just mentioned. So we are trying to identify the changes that occur when the virus infects the plant, and these encompassing changes at different levels, from the epigenome to the transcriptome, intercellular communication, hormone signaling, among other things, and also how these changes ultimately translate into developmental changes. And this is important because these changes in development frequently underlie symptom development during the infection. So these Gemini viruses are transmitted by insect vectors. And once the insect feeds in the plant and transmits the virus really has a lot to do. So there are many processes that need to be manipulated by the virus and accomplished successfully in order for this virus to establish an infection. But overall, I think we can classify all these processes in 3 main blocks. One of them would be replication. And in the case of Gemini viruses, replication occurs in the nucleus by hijacking the plant DNA replication machinery. Then, of course, there's the suppression of plant defenses. So the plant will deploy certain defense mechanisms that the virus has to overcome or inhibit. And then there's viral movement, and this includes intracellular movement and also intercellular movement, which can be short range, cell to cell, or long distance through the vascular tissue in order to, for the virus to infect systemically the plant. So today I'm going to tell you a couple of brief stories focused on how the virus suppresses defense that is deployed by the plant. So since we are interested in the molecular interactions between the virus and the plant, one of the first things that we did when my lab started in 2015 was to try to infer the interactome of the virus in the plant. And for this, we expressed tagged versions of each of the viral proteins in Nicotiana benthamiana, and we performed immunoprecipitation and mass spectrometry analysis to identify which plant proteins associated with each viral protein. And following this approach, we found a number of interesting things. And today I'm going to tell you about one of them specifically. And that is how one of the viral proteins that is called V2 interacts with a plant protein that is AGO4. So this interaction that we found in IPMS, we could confirm by immunoprecipitation. And there are 2 orthologs of AGO4 in Nicotiana benthamiana, AGO4-1 and AGO4-2. And as you can see here, V2 can interact with both of them. And we could also confirm this interaction by split luciferase assays. So in Arabidopsis, there is 1 AGO4. In Nicotiana benthamiana, there are 2, as I just mentioned. But the natural host of this virus is tomato, and it is also the agronomically relevant host. And in tomato, there are 4 AGO4 paralogs, AGO4A to D. So we cloned 3 of them. AGO4 A, B, and D, and we could confirm the interaction between V2 and these tomato AGO4s. And we could also confirm this interaction by, again, split luciferase acid. So V2 can interact with AGO4 from Solanaceae, at least from tomato and from Nicotiana benthamiana. And I will just say it now because I will come back to this later. V2 also interacts with AGO4 from Arabidopsis. But who is AGO4? So AGO4 is a central component of the plant-specific RNA-dependent DNA methylation or RDDM pathway. And this is a pathway that, as its name indicates, leads to DNA methylation. And DNA methylation is a repressive mark that occurs on cytosine residues in 3 different contexts: CG, CHG, and CHH. So this mark leads to a DNA configuration that is refractive to gene expression. Therefore, it's associated with silencing. And this pathway is supposed to defend against invading nucleic acids, which encompass also plant viruses and transposable elements. So in a nutshell, this pathway depends on the activity of 2 plant-specific RNA polymerases, Pol4 and Pol5. So Pol4 will produce transcripts that are transformed into double-stranded by the action of RDR, and then they are diced by DCL3, forming small interfering RNAs that have 24 nucleotides in size. So these siRNAs are loaded onto AGO4. And then AGO4, by base complementarity, can join the nascent transcript from Pol5, and in turn recruit the DNA methyltransferase DRM2. And DRM2 would in turn methylate adjacent DNA sequences. So we wondered whether AGO4 could be playing an antiviral role against TYLCV. And in order to test this, we silenced AGO4 by virus-induced gene silencing, or VIGS, in Nicotiana benthamiana. And as you can see on this graph, we can silence both orthologs of AGO4, AGO4-1 and AGO4-2, quite efficiently. And this silencing is specific because the close family member AGO6 is actually not affected. And when we challenge these silence plants with the virus, we can see that in the case of the wild-type virus, it can perform a little bit better. So it accumulates to slightly higher levels. But this difference is much more dramatic when we use a V2 null mutant virus that can no longer produce V2. So this V2 null mutant virus can barely accumulate in inoculated plants. But this changes when we silence AGO4, when the virus accumulates more than 3-fold more. So AGO4 plays an antiviral role, and silencing of AGO4 can partially complement the lack of V2 in a V2 null mutant virus. And this suggests that one of the main functions of V2 during the infection could be inhibiting the function of AGO4. So the viral genome can be methylated in the plant, and this is supposed to be part of the antiviral defense mechanisms. This has been described for different Gemini viruses in different hosts. You have some of the relevant references at the bottom of this slide. But we sought to determine whether we could also see this change in our experimental system. So we measured methylation of the intergenic region of the viral genome in local infection assays in Nicotiana benthamiana. And what we found is that in the case of the wild-type virus, the methylation level is really, really low, almost undetectable in most of the cases. But again, this situation changes quite dramatically when we remove V2. So a V2 null mutant virus accumulates DNA methylation in this region to pretty high levels. And this methylation increases in time, because you can see that at 9 days after inoculation, here in blue, the methylation level is higher than at 3 days in black. So the viral DNA is methylated, and V2 is suppressing this mark. So what happens if now we add AGO4 to the equation? So if we silence AGO4 and infect these plants with a V2 null mutant virus, you can see here that we reduce the level of DNA methylation basically 40%. And we can observe this in local infections, but also in systemic infections. So V2 acts suppressing the methylation of the viral DNA, and it seems to be doing so at least partially in an AGO4 dependent manner. So If V2 suppresses the methylation of the viral DNA and it interacts with AGO4, one could hypothesize that V2 could be interfering with different steps of this pathway. So one possibility would be that V2 could be interfering with the loading of the viral small interfering RNAs onto AGO4, or it could be somehow inhibiting the association of AGO4 to the viral-derived RNA or DNA, or the recruitment of DRM2. So in order to gain some insight into the specific molecular mechanism underlying this activity of V2, we first evaluated the ability of AGO4 to associate to the viral DNA in the presence or the absence of V2. And for this, we performed a chromatin immunoprecipitation or CHIP assays using a tagged version of AGO4. And we measured the binding of AGO4 to two different regions of the viral genome. Here we have the intergenic region in purple and the V2 coding region in orange. And we used an endogenous locus in the plant genome, in this case acting as a negative control. So you can see here that when we utilize the V2 null mutant, the binding of AGO4 to the viral genome increases significantly in both regions that have been studied. Therefore, we can conclude from here that V2 is interfering with AGO4 binding to the viral DNA. So we next tested whether AGO4 binding to the virus-derived RNA is affected by the presence of V2. And for this, we performed RNA immunoprecipitation or RIP experiments. And as before, we measured two different viral regions, the intergenic region and also the V2 coding region. And we used an endogenous locus as negative control. And similarly to what we have observed for the CHIP assays, the V2 null mutant shows a much increased binding of AGO4 to this RNA. Therefore, V2 interferes with the AGO4 binding to this molecule. We next tested whether V2 could be affecting the loading of the viral small interfering RNAs to AGO4. And in this case, you can see here the total small RNAs, viral small RNAs in our samples. And this total accumulation of small RNAs is not affected by the absence of V2. But when we IP AGO4 and we measure the level of small RNAs that are associated to this protein, we find that the absence of V2 does not result in an increase. Therefore, V2 is not impairing the loading of the viral small interfering RNAs to AGO4. It's interfering with downstream steps in this pathway. So something very interesting that we found was that when we were trying to see where V2 localizes in the cell, and you can see V2 fused to GFP here in green, we observed that V2 can be found in different subcellular localizations, including an association to the endoplasmic reticulum. Likely there is a little bit in the nucleoplasm, but you can observe a very clear body inside the nucleus. So what is this body? So actually colocalization with fibrillary here in blue, which is a marker of nucleolus and Cajal body, clearly indicates that this body is the Cajal body. So the Cajal body is a subnuclear body that participates in the maturation of certain nuclear RNA species and also in the formation of small nuclear ribonucleoprotein complexes, which can be involved in different processes, for example, transcription, splicing, or telomere maintenance. So this was very interesting because in 2006, it was described that AGO4 and some other components of the RDDM pathway in Arabidopsis concentrated in the Cajal body. However, the biological relevance of this localization has so far remained elusive. So we checked and we found that indeed, also in the case of Nicotiana benthamiana AGO4 and tomato AGO4, we can see a clear accumulation in the Cajal body. And even more interestingly, when we checked where the interaction between V2 and AGO4 is happening in the cell, we found to our surprise that this interaction seems to be pretty specific for the Cajal body. So you can see here V2-V2 homotypic interaction as a positive control. This interaction happens in the nucleoplasm and in other subcellular localizations, also in the Cajal body. But the interaction between V2 and the AGO4 proteins from Nicotiana benthamiana or from tomato occurs almost exclusively in the Cajal body. So V2 interacts with AGO4 in the Cajal body, and this interaction in this very specific location is sufficient to affect DNA methylation of the entire viral population. So in order to determine whether Cajal body localization is really relevant for the function of V2 in the suppression of viral DNA methylation, we used a little trick. Because what I showed you before was a V2 GFP fusion protein, and you could see that it does localize to the Cajal body, as you can see also in this image here. However, if we make an N-terminal fusion of this protein, that is GFP-V2, now this protein no longer can be found in the Cajal body, but otherwise shows an almost indistinguishable subcellular localization. And this is just a quantification to show you that this presence or absence in the Cajal body is pretty consistent and happens, as I just told you, in almost 100% of the cases. So then we decided to do a complementation assay. So again, we measured the methylation of the intergenic region of the viral genome in local infection assays using wild-type virus or a V2 null mutant virus, and then complementing this V2 null mutant virus with V2 GFP, which localizes to the Cajal body, or GFP-V2, which does not. So first you can see that we can detect in this case a little bit of methylation in the wild-type virus, but this methylation, this is here in black, but this methylation in dark blue is much, much higher in the case of a V2 null mutant virus. However, this level of DNA methylation can be partially decreased if we complement this V2 null mutant virus with a V2 GFP version of V2, which again localizes to the Cajal body. However, the GFP-V2, which does not localize to the Cajal body, cannot really complement this accumulation of methylation in the intergenic region of the viral genome. So GFP-V2 does not suppress a methylation of the viral DNA. However, even though I'm not showing this to you here, I can tell you that it can still interact with AGO4 in co-IP assay. So GFP-V2 has the ability to interact with AGO4, but in the plant cell does not find it, most likely. And then we decided to take a complementary approach to, again, reassess the relevance of the Cajal body for the viral infection, and in particular for the methylation of the viral DNA. It is known that one of the main components of Cajal bodies, probably scaffolding component, is coiling. And if we silence coiling, then we get cells that are devoid of Cajal bodies. So we silence coiling again in Nicotiana methamiana again by using VIX. In this case, we got a partial silencing of roughly 50%, but this was sufficient to make the Cajal body disappear. So you can see here, this is a control plant infected with the empty vector virus, and we can clearly distinguish the Cajal body, but this is lost in a plant where coiling has been silenced. And if we challenge these silenced plants with the V2 null mutant virus, you can see that the methylation of the intergenic region is basically halved. So you have the V2 null mutant virus in the dark pink, and in the case of the coiling silenced plants in light pink, and the level is roughly 50%. So either Cajal bodies or coiling itself are required for full methylation of the viral genome, and therefore we are establishing a direct connection between the Cajal bodies and antiviral DNA methylation. So based on these results, this is the model that we propose. So we think that when the Gemini virus is infecting the cell, there is production of viral small interfering RNAs, which can be loaded onto AGO4. And then AGO4 can be recruited to the viral DNA through interacting with the virus-derived RNA. And then it will recruit most likely DRM2, and then the viral genome will be methylated and therefore silenced. However, the virus can produce V2, and V2 can interact with AGO4. This interaction happens specifically in the Cajal body, and then it will be interfering the recruitment of AGO4 to the viral genome. Whether this process here of viral DNA methylation occurs in the Cajal body or not, this is something that we currently don't know, and is something that we are investigating at the moment. But clearly there is something going on with the Cajal body. So I just showed you that, well, it looks like there is a connection between Cajal body and antiviral silencing, but also when we infect cells with our virus, we see that the Cajal body in some cases starts apparently multiplying. So this is a dynamic process, and it's also something that we are currently trying to understand better. And something else that we are trying to do now as a second part of this project is to evaluate whether V2 can have a genome-wide effect on DNA methylation. And for this, we have moved to Arabidopsis. As I mentioned at the beginning, V2 can also interact with tug-of-war from Arabidopsis. So we have generated transgenic Arabidopsis lines expressing V2, and we are comparing the methylome of these transgenic plants to that of an tug-of-war mutant. And as you can see here, there is a significant overlap in differentially methylated regions. So if we look at some of these hypomethylated regions that are in the overlapping sector of this Venn diagram, then you can see that these loci behave similarly in the V2 transgenic lines and in the tug-of-war mutant in this hierarchical clustering analysis. And even more interestingly, while tug-of-war acts predominantly on transposable elements, this is not the case for V2. So V2 seems to affect mostly genic regions, and this is true for the differentially methylated regions in the V2 expressing plants alone, but also for the regions that are found in the overlap between V2 and tug-of-war mutant. So V2 is indeed affecting the Arabidopsis methylome, and it is doing so in a manner that is partially similar to that of an tug-of-war mutant, but preferentially seems to affect gene sequences. And how this is really regulated, we don't know, and this is something we are also trying to look into. And what you can see on your screen now is Shanghai, and more specifically the Pudong area. So you can see the well-known skyline and the iconic TV tower, the Oriental Pearl Tower. The river that you see there is the Huangpu River, which divides the city into the east part, that is Pudong, and the west part, that is Puxi. And now that hopefully I have regained your attention, I can switch gears and move on to the second part of my talk, where I am going to tell you a little bit more about a different viral protein. So this other viral protein is C4. So C4 is the smallest protein encoded by TYCV. It's only 10 kilodalton in size, and it's encoded by this open reading frame in the red box, which is completely embedded into another gene, just in a different frame. So when we express C4 in the plant cell, we can find it associated to the plasma membrane, as you can see in this image, and also in punctate structures here on the image on the right. And these structures are actually plasmodesmata. So these are the channels that join adjacent cells. So if you look carefully to the picture on the right, perhaps you can see that C4 is not only at the plasma membrane, it's also somewhere else. So C4 is also in chloroplasts. And actually this dual localization at the plasma membrane and in chloroplasts should not really come as a surprise, because if we look at the protein sequence, we can identify two targeting signals which are overlapping. So there's a meristylation site at the end terminus, and meristylation is a lipidation that tethers proteins to membranes. And there's also a chloroplast transit peptide that spans roughly half of the protein and would be targeting the protein to this organelle. So as we could predict, perhaps, if we mutate the meristylation site, so C4 can no longer be meristylated, then it can only be found in chloroplasts. And we can do this by mutating the glycine in position 2, that is the residue that is meristylated. So the end meristylation site is required for the plasma membrane localization of C4, and this plasma membrane localization overrides chloroplast localization. And actually, we could identify this meristylation in vivo by mass spectrometry analysis. And we could also see that if we disrupt meristylation, and we have done this in two different manners, we have silenced the meristylate transferase, that is NMT, or we have treated the plants with a chemical benzothiazole, which is an inhibitor of this enzyme. And in both cases, we get a similar effect. So C4 disappears, or at least largely disappears from the plasma membrane and starts accumulating in chloroplasts. So C4 is merystolylated in planta, and this merystylation is required for its plasma membrane localization. So this means that this tiny viral protein can be found in two subcellular locations. It can be found at the plasma membrane, and it can be found in the chloroplast. And this suggests that this protein must have at least two different functions during the viral infection. So we found this very intriguing, and we decided to try to learn more about what this protein is doing in the plant cell. So I'm not going to tell you a lot about what C4 is doing at the plasma membrane, because part of this work is already published. And you can just take a look. But basically, in a nutshell, C4 interacts with a receptor-like kinase that is BAM1, and also its closest homologue BAM2. And this interaction, of course, happens at the plasma membrane, but it's particularly strong at plasmodesmata that you can see here as bright dots. And we know they are plasmodesmata because they co-localize with aniline blue staining, which stains callose. And basically, considering the results that we got, we came up with this model according to which BAM1 and BAM2 are sitting at plasmodesmata, and they are somehow promoting the cell-to-cell movement of RNA silencing, which is considered the main antiviral defense in plants. But when C4 is there, then C4 interacts with the kinase domain of these receptor-like kinases, and it somehow interferes with this particular function of the RLKs. How exactly C4 is doing this, and what is the molecular mechanism by which BAM1 and BAM2 can promote the spread of silencing is something that we don't fully understand yet, and we are currently working on that. And I have to say, though, that at the plasma membrane, C4 is most likely doing additional things, because we found that C4 does not interact only with BAM1 and BAM2. It interacts with a number of receptor-like kinases, not every receptor-like kinases, but with a broad range of them. And also, if we express C4 in its wild-type form in Arabidopsis, you can see that this leads to very strong developmental defects. However, these strong developmental defects require the presence of C4 at the plasma membrane, because if we express the non-merystylable mutant that, if you remember, goes to chloroplasts only, these plants look pretty happy. So C4 is doing something at the plasma membrane that is leading to these developmental abnormalities, but we do not think this effect is linked to BAM1 and BAM2, because a BAM1, BAM2 double mutant do not display phenotypes that are similar to the ones displayed by transgenic C4 plants. And now I would like to spend the remaining time of this talk to tell you a little bit more about what C4 is doing in the chloroplast. So we got a hint at what C4 might be doing in the chloroplast when we observed that in basal conditions, C4 is mostly localized to the plasma membrane, but this situation completely changes when the cell is infected by the virus. So when the virus is there, C4 starts being depleted from the plasma membrane, and it starts accumulating in chloroplasts. And we know that one of the things that happens when the virus is infecting the cell is that the plant can somehow perceive the presence of the virus, and it can trigger a certain level of defense. So we wonder whether it could be this activation of defense that is leading to the relocalization of C4. So we decided to test what happened if we activated defense in a completely different manner. And we did this by treating with the immunogenic peptide FLAG22, which is derived from bacterial flagellin. So we treated plants with FLAG22, and to our surprise, we found that C4 behaved in a very similar manner to what it does when the virus is there. So C4 started disappearing from the plasma membrane, and it started accumulating in chloroplasts. And this relocalization of C4 that we can observe visually under the microscope, we can also see biochemically if we purify chloroplasts and measure the level of accumulation of C4. So as you can see here in the red square, after FLAG22 treatment, we have a much higher accumulation of C4 in the chloroplast. And you can see that the size of this C4 protein is smaller than the one that we can see associated to the plasma membrane, because upon import to the organelle, the transit peptide is cleaved. And this does not happen only if we activate defense by treatment with FLAG22. We can also use other pumps, and we can also use damage-associated molecular patterns like PEP1, which is an endogenous plant peptide, and we observe a similar thing. So as you can see here, again, we treat with PEP1, and C4 starts accumulating in the chloroplasts at the expense of the plasma membrane pool. But what do chloroplasts have to do with defense? So actually quite a lot. So it is well known that when there is perception of a biotic threat, which frequently happens at the cell surface, this information is somehow relayed to chloroplasts. And then chloroplasts in turn will communicate with the nucleus through retrograde signaling. And this will lead to transcriptional reprogramming. And this transcriptional reprogramming affects genes that are required for the biosynthesis of the hormone salicylic acid, which is one of the main defense hormones. So there is a biotic threat perceived. This information reaches the chloroplast. The chloroplast tells the nucleus we need to synthesize salicylic acid. So then we wonder whether C4 could be interfering with this pathway. And in order to test this, we moved on to Arabidopsis, and we generated transgenic Arabidopsis plants expressing this non-myristic label version of C4. So these plants express C4, and this C4 goes to the chloroplasts only. We treated these plants with a pathogen-associated molecular pattern FLAG22 to activate defense. And then we measured the expression of the SA biosynthetic gene ICS1 or the SA responsive gene PR1. And as you can see here, compared to the control plants, the expression of these marker genes is decreased in the C4 expressing lines. And actually, the accumulation of salicylic acid itself is roughly halved in these plants. And this partial suppression of SA accumulation seems to be relevant for the viral infection, because plants that are depleted in SA, for example, Naji plants, are more susceptible to the viral infection, so the virus accumulates better. And even more interestingly, this leads to a partial complementation of the lack of C4 in a C4 null mutant virus. So chloroplast localized C4 can suppress SA-mediated defenses, and this suppression of SA-mediated defenses seems to be one of the main roles of C4 during the infection. So next we wondered, if C4 is doing this, how is it doing this? And we reasoned that most likely it would be through the interaction with some plant protein. So we attempted to define the interactome of C4 in the chloroplast, again by IPMS analysis. And we found one interactor that we deemed as particularly interesting, and that is a calcium sensing receptor, or Cas. So the interaction between C4 and Cas, we could confirm by biopsy, as you can see in this image, and also by co-immunoprecipitation. And the reason I say that we deemed Cas as particularly interesting is because Cas had already been described as participating in retrograde signaling in defense, in particular in the response to FLAG22. And it had been shown to activate SA biosynthesis and downstream responses. So we thought, okay, maybe C4 is interacting with Cas and inhibiting its function, and therefore it's having an impact on the downstream responses. So we checked a number of defense-related readouts in the transgenic plants expressing the chloroplast version of C4 and compared them to a Cas null mutant plant. And as you can see, they show phenotypes that are very similar. So these plants are all more susceptible to infection by the bacterial pathogens Pseudomonas syringae, Pathobacteremia DC3000. They also show defects in the transcription response to FLAG22, which show a statistically significant overlap. And of course, if we look specifically at Cas-dependent genes, these genes are downregulated both in a Cas mutant and in the C4 expressing plants. So C4 interacts with Cas, and the plants expressing C4 and the plants mutated in Cas show phenotypes that are very similar, supporting a potential inhibition of Cas by C4. And this potential inhibition of Cas would actually have an impact on the viral infection. So an Arabidopsis Cas mutant is more susceptible to the viral infection, as you can see here, and it partially complements the lack of C4. And we observe a similar phenotype if we silence Cas in Nicotiana benthamiana or in tomato by VIX. These Cas silence plants are more susceptible to the viral infection and also show a partial complementation of the lack of C4. So basically, we came up with this model to explain the role of C4 in viral pathogenesis. So C4 is produced in the cell. It is myristylated co-translationally and therefore goes to the plasma membrane first. And at the plasma membrane, it interacts with BAM1 and BAM2 and inhibits the cell-to-cell movement of RNA silencing. But upon activation of defense, which is something that happens when the virus is there, then C4 can somehow relocalize to the chloroplast, and there it interacts with Cas and potentially inhibits its function, therefore suppressing assay signaling. So in the end, C4 indeed has two different roles, one on the plasma membrane and one in the chloroplast, and of course both of them end up promoting the viral infection. So we think this is a very nice example of how subcellular compartmentalization can underlie multifunctionality of a viral protein. And something that I would like to draw your attention to is that we have observed that when we trigger the relocalization of C4 by activating defense, for example, by treating with FLAG22 or with PEP1, we can see that even though C4 is depleted from the plasma membrane, it seems to be retained at plasmadesmata. So now you can very clearly see the accumulation of C4 in these structures. And this suggests that even though C4 moves to the chloroplast when the plant perceives the presence of the virus, it can still remain at plasmadesmata, and therefore it has the chance to perform both virulence activities at the same time. So when we were at the beginning of this project and we were trying to learn a little bit more about myristoylation and chloroplast transit peptides and these targeting signals, we stumbled in the literature upon something that was very intriguing, because we found that it had been published that one protein from Arabidopsis, and that is calcium protein kinase 16 or CPK16, contained the same two targeting signals that C4 has, an N-myristoylation motif and a chloroplast transit peptide. And similarly to C4, if the wild-type version was expressed, then this protein accumulated at the plasma membrane, but a non-myristylable mutant version of this protein would start accumulating in chloroplast. So then we wondered whether this CPK16 protein may behave like C4. So we treated tissues expressing CPK16 GFP with FLAG22. And indeed, we found that now CPK16 moved from the plasma membrane to chloroplast. And again, we can see this visually under the confocal microscope, but we can also detect this change in Western blot analysis after chloroplast purification. So then, of course, we wanted to assess whether CPK16 is having a role in defense from the chloroplast. So for this purpose, we generated transgenic Arabidopsis plants overexpressing CPK16 in its wild-type form, or in its non-myristylable form that goes to the chloroplasts only. And as you can see here, these plants look pretty happy. There's no developmental effect of overexpressing any of the versions of CPK16. But when we treat these plants with FLAG22 and now measure the expression of the SA biosynthetic gene ICS1, you can see that there is a mild increase in the plants that overexpress CPK16 in its wild-type version, but this increase is notable in the plants that express the CPK16 protein that accumulates in the chloroplast only. So CPK16 from the chloroplast can increase this expression of the SA biosynthetic gene. And in agreement with this, these plants that overexpress CPK16 in the chloroplast are more resistant to Pseudomonas, and they are also more resistant to the viral infection. So chloroplast localized CPK16 is acting as a positive regulator of defense. So this combination of the two targeting signals that we found in C4 exists in plants. So there is at least one case. So then we wondered whether maybe it would be more prevalent than we initially thought. So we decided to screen the Arabidopsis predictive proteome looking for plants with these two signals together. And to our surprise, we found 73 of them. And then we did the same thing with tomato and rice. And we also found considerable numbers of proteins with these two signals together. And there was an overlap of 26 proteins among the 3 species. So we took this subset of proteins and performed a functional enrichment analysis. And then we found that indeed there are overrepresented functional geoterms, which include salicylic acid mediated signaling and systemic acquired resistance. So it looks like there is a conserved core of defense regulators in plants that contain these two signals together. And interestingly, the combination of these two signals can be found throughout the plant kingdom. So it's already present in Chlamydomonas, and we can also find it in Mercantia and Fiscomitrella, and then of course in vascular plants. So if this combination of targeting signals exists in plants, and we found it in C4, then we reasoned other plant viruses might have evolved to incorporate these targeting signals as well. So we screened the proteins produced by plant viruses, and we found indeed more than 400 viral proteins with these two targeting signals together. So I have to say most of these proteins are C4 orthologs in different Gemini virus species, but there are also proteins that are completely unrelated and that are produced by RNA viruses. So independently evolved viral proteins have somehow convergently acquired these two signals. And then we did the same with a completely unrelated pathogen, a bacterial pathogen, Clostridium solanacearum, and we screened the entire bacterial proteome. And we found 2 proteins in the entire proteome with these two signals together. And possibly not very surprisingly anymore, these 2 proteins are known to be effectors. That is, they are proteins that are produced by the bacterium and are injected into the plant cell in order to promote bacterial virulence. So I don't have time to go too much into detail, but we have done some preliminary characterization of some of these pathogen-encoded proteins with the coexistence of these 2 signals. So you can see here proteins from 3 independent viral families, Gemini viruses, but also 2 RNA viruses. And you can see the subcellular localization of these proteins in their wild-type version first, and below in their non-myristoylable mutant version. So you can easily see that in all cases, these proteins appeared as associated to membranes to some extent at least. And when we mutate the myristylation site, then they invariably appear in chloroplasts. So these signals are indeed working. And at least in some cases here in these 2 Gemini virus species, we can see a relocalization following a FLAG22 treatment. So indeed, these proteins can move when we activate defense. And perhaps even more interestingly, if we express the chloroplast localized version of these proteins in Nicotiana benthamiana, this is sufficient to hamper the expression of ICS1 or PR1, these SA biosynthetic and responsive genes, when we treat with FLAG22. And in all cases but one, expression of the chloroplast localized version of these proteins renders the plants more susceptible to infection by Pseudomonas. So in a nutshell, proteins that have independently evolved in different plant viruses contain these 2 targeting signals together and can suppress defense from the chloroplast. So basically, our results suggest that in plants there is a pathway that connects the plasma membrane and chloroplasts and activates defense upon perception of a biotic threat. But throughout evolution, this pathway has been co-opted by pathogens, and these include at least viral pathogens and bacterial pathogens. And these pathogens have evolved to produce proteins that have acquired these 2 targeting signals and now can move from the plasma membrane to the chloroplasts when their presence is perceived. And from the chloroplast, they can suppress the activation of downstream defense responses, therefore promoting virulence. And with this, I'd just like to thank the people who did the work. I am really lucky to be working with a fantastic group of highly motivated and talented young scientists. And I would like to thank our collaborators. I have listed here only the ones for the C4 related projects. I would like to thank funding agencies, of course, and you for your attention. And I would be happy to take questions if there are any.
Okay. Rosa, thank you very much for this fantastic seminar. And we will start now the question. So we have two ways to do this. One is you can write your question in the chat, and Rosa can read it and just answer to your question, or alternatively you can just tell us that you want to ask your question using the microphone, and we will activate your microphone. So, but if you use the chat, it will be easier. Okay, so Aaron is saying here that you can activate your microphone without any problem already. So questions?
Can I make a question? Sorry.
Yeah, sure.
Rosa, maybe I missed something. I'm Maria Ángeles Ayllón. Maybe I missed something, but what happened if you delete the C4 gene in the virus? Is it infectious, or I don't know?
Yeah, so no, actually, I think maybe I didn't show this. So we cannot really delete the entire gene because C4 is embedded inside another gene that is the rep gene, and that is essential for the virus. So we cannot remove the entire sequence, but we can either remove the start codon, or we can introduce a mutation to have an early stop codon, and then the virus can no longer produce C4. So this C4 null mutant virus can really barely infect the plants. So if you can inoculate tomato, Nicotiana benthamiana, Arabidopsis, anything you want, the virus can barely do anything. So C4 is essential for infectivity.
Okay, thank you.
Okay. So,
may I ask a question?
Sure. So
I am Fernando García Arenal, and in this last part of your talk, in which you find proteins that have both the methylation and the chloroplast transit domain in different genera of viruses, you put a couple of examples, but I was not able to see if these proteins have the same function in the virus life cycle. I mean, are they movement proteins in all the case, or are there different proteins according to the genus?
Yeah, they are different. So what we have, I mean, in the case of Gemini viruses, it's all C4 proteins.
Yes.
And then we have a capsid protein, and then we have a P3 protein. So in principle, they are functionally unrelated in theory.
Okay. Okay. Thank you.
Emilia Lopez Solanillo.
Yes, thank you, Rosa, for this exciting talk. I have two questions. The first one is it known if the DRM2 protein also localizes in Cajal bodies?
As far as I remember, this was not, I think, described. So it has been shown for NRP1 that it localizes in the Cajal body. But then in a later paper, they showed that actually this only happens in a certain percentage of the cells. So it's not actually the main situation. So tug-of-war seems to be the RDDM component that is most frequently localized in the Cajal body, but not other components. So as far as I remember, DRM2 has not been demonstrated, but I would not put all my money on it.
Okay. And well, my other question regards to the second part of your talk, which is, for me, is very exciting. So do you know something, or do you have any clue about how the relocalization of C4 take place? I mean, is it using the vesicle traffic system of the cell, or?
Yeah, this is the $1,000,000 question, really. We don't know yet. We don't know yet. So we do see structures that look like vesicles containing C4 after we activate defense, or when the cell is infected with a virus. So it is very possible. We don't know how C4 would be, I mean, at some point C4 has to be detached from the membrane in order to enter from the, through the TikTok complex into the chloroplast. So how this happens, we don't know yet. So in animals, it has been described that phosphorylation of a myristoylated protein can be sufficient for the protein to be released from the membrane. And we know C4 is phosphorylated. So this would be one possibility. Myristoylation is supposed to be irreversible. So in that case, the myristoyl group should be somehow, like the sequence containing these sites should be somehow cleaved, or perhaps actually is not irreversible, because apparently this demyristojlase activity has been found in bacteria. So maybe this is something that exists but is yet to be described. So we really don't know, and these are the main possibilities we are now exploring. So we have one mutant version of C4 that seems not to move anymore from the plasma membrane. So we are trying to take advantage of this to try to gain some further insight. And something else that we are doing is we can use a little trick, because we can silence one component of the TikTok complex, and then C4 no longer gets imported into the chloroplast, or it gets imported but with very low efficiency. So if we express the wild-type version of C4, silence this TikTok complex, and then trigger relocalization, then we will have this released C4 protein accumulated in the cytoplasm. So we can now try to identify which changes happen to this protein by mass spectrometry analysis. So this is our hope. So these are more or less the approaches that we are following now. But yeah, right now we have hypotheses. We don't have many clues yet.
Okay, thank you. Thank you, and congratulations for your nice results.
Thank you very much.
Rosa, there is a question of Nacho Rubio. It is a very nice talk. I was wondering whether you checked to force C4 to stay in the membrane, and what is the impact on defense, for instance, by removing the chloroplast transport signal. Thanks. Okay, so this is a little bit tricky. First, because the chloroplast transit peptide is not really conserved in sequence, so it's not easy to decide what to mutate to impair its functionality. But more importantly, actually the region that interacts with BAM1 is precisely the chloroplast transit peptide in C4. So if we were to remove this or mutate this, then we would be affecting what we consider the main function of C4 at the plasma membrane, and therefore this would be very difficult to it will be very difficult to really evaluate the relevance of each of the subcellular localizations. So for now, we can send C4 specifically to the chloroplast, removing it from the plasma membrane. This is okay. And we can see that actually you need C4 at the plasma membrane for full virulence. We cannot do the opposite, except for the case of this one mutant version of C4 that I just mentioned. So we have this mutant version. It doesn't seem to move from the plasma membrane, but the interaction with BAM1 and BAM2 and some other plasma membrane interactors doesn't seem to be affected. So this could be potentially a good tool. So this is something that we are looking into at the moment. Okay, thanks. There is another question of Nicolas Cecchini, but there are several questions. Maybe Nicolas, can you ask the question directly by using your microphone?
Okay, can you hear me?
Yeah.
Okay, very nice talk, Rosa. I think you answered some of my questions about how can this transit peptide that is lipidated be recognized by the TikTok complex, for example, or maybe if it can exist some membrane targeting between plasma membrane and plastid membrane or something like that.
Yeah, so we really don't know. The chloroplasts seem to be, when we activate defense, when we express C4, they seem to somehow get closer to the plasma membrane, but we have not really quantified this, so I'm not 100% sure. It could be, as we mentioned, through vesicle trafficking, but as you said, the we would expect that C4 needs to be demyristoylated somehow. So the myristoyl group should not be there for efficient import. I am not an expert in chloroplast import, but we are collaborating with a group of Chang-Hong Kim here in PSC, and his research is focused on chloroplasts. And he agrees that the myristoyl group should not be there. He doesn't envision how the protein, which has to be denatured in order to pass through these channels, can have a lipid group attached and still be efficiently imported. So somehow this should disappear, but how exactly, we don't know yet. So
yeah, it is possible that you are seeing like two different mechanisms, like when you have the mutant version, it's just the classic transit peptide, but the other it's using another something different.
you mean in which? So when we
the wild type version is different compared to the mutated version, the trafficking, the targeting to the plastids.
Well, we cannot fully rule that out, I mean, in the case of the mutant, the non-myristylable mutant, it is never myristoylated. So in this case, it should be easy, right? It can be imported directly. In the case of the wild type version, it has to go to the plasma membrane first, and then we have the problem. So yeah, we are hoping that this, if we silence the TikTok complex and then purify the, let's say, activated population of C4, then we should have a chance at spotting differences. So for example, if the N-terminal part is cleaved, then we should see that this part is missing, right? Or if there's very heavy phosphorylation, or also, you know, for in the case of myristylation, like we should be able to see if now myristylation can no longer be detected.
Yeah, exactly. And one last thing, did you see any mitochondria in some of your proteins, considering the similitude between the transit peptides? Just asking.
Yeah, no, this is, I mean, it's definitely a good question. So we did check, and we do not see mitochondria. We do not see C4 in mitochondria. So we can find some other viral proteins in mitochondria, but not C4. So that this transit peptide seems to be pretty specific in this case.
All right, amazing talk. Thank you.
Okay.
Thank you.
Some additional questions. So, Rosa, I have a couple of questions. One is related to I was surprised that the C4 is interacting almost with all the RLKs that we know that are involved in defense. So there is a specific motif for this interaction that is conserved in all the RLKs. So it's just, I mean, all this PRR, or it's just a matter of the localization of the C4 in the membrane where is also where these PRR are also located.
So we think most likely there has to be some degree of specificity because it does not interact with all of them. So for example, you saw in the diagram, it doesn't interact with BAC1 for some reason, but it can interact with FLS2 and with BAM1 and others. So in the case of BAM1, we know that the interaction occurs with the kinase domain.
Okay.
So it's possible that there is some, you know, that C4 has evolved to target some specific RLK, and then as a, you know, side effect, it can interact with others because the 3D structure is similar. But we don't know because we have not characterized this more in depth. This was just an observation, and we thought it was important to keep this in mind.
Okay. My second question is related with your funnel scheme just before the slide that your group. So because clearly this, I mean, I guess it's very exciting that this virus and some plant pathogen may have co-opted this mechanism of signaling of like you have described for the C4. But regarding how this C4 is interfering with the defense activation is just by interacting with the, for example, CPK16. So how is the mechanism of blocking? So because for me this is not totally clear.
Yeah, in the case of C4, what we found is that C4 interacts with this Cas protein, and Cas is essential for retrograde signaling. And of course, we have also reported the interaction, and then the phenotypes are similar. So we think this interaction leads to a suppression, inhibition of the Cas activity. But how this would be happening at the molecular level, we don't know yet. We have tried to look into post-translational modifications and some other things, but it's still unclear. But we think it's through the interaction with Cas. We have not found an interaction between C4 and CPK16. It doesn't mean it doesn't exist, but we have not seen it.
And CPK16 is interacting also with Cas or not really?
This we have tried, and the result is negative, but again, it's just one negative result. So it may or it may not. I mean, the nice thing is that now we have a bunch of proteins that go to the chloroplast and do different things. Some activate defense, some repress defense. So I think we could use these different proteins as probes and try to identify interactors, and perhaps we can identify different components of the pathway, because we would not expect that all of them are targeting exactly the same protein.
Yeah. Okay, thank you very much. So any further question for Rosa? I think this was the last question. No more questions. So if this is the case, no more question. Again, thank you very much for this fantastic presentation, and congratulations for your great result and your milestone in this research center in Shanghai. And thanks again for accepting our invitation to be the first speaker of this online seminar series of the CBGP. And thank you to all the attendants for joining us in this first experience. And I guess it was a very positive experience. And hopefully we will have the chance to have a more tight interaction in the future once this situation with the COVID is overpassed. And Rosa, just keep in mind that we just have a commitment with you just to pay you a lunch, a Spanish lunch, a good Spanish lunch, once we have the opportunity to have to be together. Okay, so
I will accept a rain check. So
okay, so we will.
I write it down.
We will arrange for the next time. Okay.
Okay. Thank you very much for the invitation.
Thank you very much to all of you, and we will see all of you the next week for the next seminar. Bye bye.